and he like hearts my texts. He's the only person I know.
I think a lot of people use the heart function.
Well, why would you not just have a thumbs up? Like
You get an option.
I have a sentence. The nanotubes turn out to be the cellular bilayer, just from one cell pushing into the other. And I get a heart on that. Why not a thumbs up?
It's a more emotional response than a thumbs up. A thumbs up is like, I heard you. Sounds good. See you at the coffee shop at 10 a.m.
I think I actually kind of see it, right? Like a thumbs up is kind of like dismissive in a way. Like,
yeah.
Good for you.
Yeah.
Whereas a heart is like, cool.
Yeah. Like I understand.
Okay.
Yeah.
Okay.
Hi, my name is Linnea.
I'm Bezia.
And I'm Linnea. And this is episode number 25 of the Lemma Journal Club.
What is the Lemma Journal Club?
The Lemma Journal Club is the show where we read every paper to tell you if you need to read it. You probably do.
We have a terrific paper for you today. We are talking about a paper entitled Combining Sophisticated Fast FLIM, Confocal Microscopy, and STED Nanoscopy for Live Cell Imaging of Tunneling Nanotubes. It's by a bunch of French people in the journal Life Science Alliance. Linnea, had you heard of this journal before I chose this paper?
No, my very first question on my list of many questions for you is, what is this journal?
I really think this is going to be one of our best episodes. I think the major problem with this episode is how much there is to talk about. So Life Science Alliance is, I think, an open access version of, EMBO. This is a real journal. This is a weird journal, as shown by this very, very weird paper.
Very weird paper. Okay, yeah. So first question, why did you pick this paper?
Yeah, so I wanted to talk about tunneling nanotubes. I made the mistake while we were at March meeting of talking a little bit about my own research to our friends. And then I get...
Mistake.
Well, I just get stuck in it and I feel self-conscious and I'm not like really giving a talk or anything, so I can't do my actual research justice. And I talked a little bit about some of like the hypotheses I have that maybe it is possible that there are mitochondria migrating across a tissue during tissue development. Maybe in certain cases, while an organ is forming or while an organ is formed, mitochondria migrate across that organ. And I speculated that there are ways for that to happen, such as tunneling nanotubes, where mitochondria move across these tubes from cell to cell. Noah and Siraj stopped me, and they were like, explain the nanotubes, and they were quite aggressive about it. And I was like, I actually looked this up like 3 or 4 years ago. I don't really remember anything about it. And they were like, can you at least specify, is this like a monolayer or a biolayer or like a nanopore? Like, what exactly are you describing? And so I thought it would be good to do some heavy research on these nanotubes.
Okay.
And to really have like good canned answers for what they are if I'm starting to think about them. And I tried to study these at one point early in my postdoc and felt that I didn't have the technology necessary to study these. Why did I pick this specific paper? I wanted a paper with beautiful videos. And
You did that.
I did that, yeah.
Yeah.
I wanted a paper that wasn't trying to sell me on the function of nanotubes.
Okay.
So the vast majority of papers about nanotubes are jumping over trying to understand what they are and going straight to like clinical applications like, oh, then maybe this has to do...
I feel like maybe this paper landed too close.
Absolutely. Absolutely. But
Like didn't
this
even
paper,
attempt the jump.
This paper is essentially just raw data, right?
Yeah.
But like there are many papers that you can read about these tunneling nanotubes where they're trying to go straight into some critical function that they have in biology.
Okay.
And I think that this is an interesting thing about biology that I'm all of a sudden thinking about, is you could find the most fascinating thing in biology. You could find a new organelle, let's call it the mesochondria. The mesochondria could be in the cell. It looks like a spiky ball. It's doing something really weird. It's shuttling back and forth between every mitochondria and the cell nuclei. And you could show that. And if you didn't have a function for it, if you could get rid of the mesochondria and the cell was functioning just fine, most biologists would not give a shit.
Yeah.
And this is super different than I think physicists, where physicists are really interested in what is the physical world, what exists, right? So when you show that a new type of matter exists to a physicist, they're not like, okay, but what can I make out of it, right? They're like, oh, that's so cool, a boson, wow. Biologists are immediately like, what is the function? And this is why you study what you study, or this is a great thing about the pyrenoid, that it has a very clear function. So I think a problem for liquid phase separated organelles is that it's not so clear often if they have any super important function. And this has been a big fight for the past 20 years of show that some of them have really important functions so that the biologists really care. I've never understood why this is a fight. Who cares? They're part of the cell. Our job is to learn how all the parts of the cell work. But that doesn't seem to be true. Our job is to learn what is the most critical thing in the cell.
Yeah, I guess I would push back against this a little bit in that I do think function is interesting and tells you more about what exists and how it interacts with everything else.
Well, there's two things. One, when the biologists mean function, they usually mean critical function. Not does this make the cell work better or worse or healthier. They mean, does the cell die?
Well, I think that's because that's where we are with biology. It's like we've got, hammers for our
tools. Yeah.
We don't have the precision to ask more nuanced questions of like, how much does this help?
But it makes it so that
I do hear that what you wanted from this paper was to not be hit over the head with the function, the speculative function of these TNTs.
Well, so the problem with the TNTs is that similar to liquid phase separated organelles, they're a very, very recent discovery. They came out around the same time, and they came out for the same reasons that, microscopy got to this point where it could do this resolution live and you could start to see some new things. And this is another thing that people saw. And unlike liquid phase separations, which then blew up into this huge field where people are trying to figure, these tunneling nanotubes have been more or less ignored.
Yeah.
There's about 3 or 4 papers on them each year. The papers are super not into the physics or the hard molecular biology of how they work. They're mostly electron microscopy images of these. And the more modern ones are, I guess, trying to quote unquote revive this discovery by saying, hey, look, it's critical for these cardiomyocytes. It's critical for these cancer cells communicating with each other, because they've already gotten shoved to the sidelines of, sure, this is a thing that cells do, but it doesn't seem absolutely necessary for cell life, so we're not going to study it so much. And the more recent papers then are too focused on its function. And what I really wanted to understand was what the tunneling nanotubes literally were. Right? I really wanted to understand them from a molecular biology standpoint. So that's why I picked this paper.
Interesting.
Is that, when you make this beeline for function, things get a little bit weedy. And people are doing this for reasons that I think are not truly scientifically driven. That in a perfect world, I would say the vast majority of biologists should be studying how the biology works and trying to really shut out this question that everyone asks you of okay, but what can you do with that medically? Because this beelining for what are the drugs that you can use, people should do that, sure, but that's medicine, right? That's not science, right?
So with that in mind, what are TNTs and why do they form?
I did a tremendous amount of outside research while reading this paper. So tunneling nanotubes are extrusions of a cell that have essentially poked in to another cell. They are membrane bilayers, right? So the outside of the nanotube is the membrane bilayer of a normal cell, and the tube is filled with cytoplasm. So it is essentially a bridge connecting two cells, and now the cells share cytoplasm and materials.
So it emerges from one cell.
Yeah.
And then it finds another cell and creates some sort of membrane junction to start exchanging things.
Yeah, and I think you want to be careful about the word junction here, because a junction is normally a different thing. Whereas here, it's really created a continuous membrane so that the topology of the cells are now that, in some sense, they are one multinucleated cell, right? The thing that I found interesting that I didn't already know is that there are kind of 2 subcategories now of tunneling nanotubes. There's tunneling nanotubes that are thicker, that contain actin, microtubules, and vimentin. And there are tunneling nanotubes that are thinner, that contain just the actin. I always thought that the mitochondria were using Miro1 linked to a kinesin to ride these microtubule networks. It turns out that Miro1 has a second mode where it can link to myosin motors, and Miro1 can also surf along on actomyosin. So presumably mitochondria can move through both types of these tunneling nanotubes.
And the only difference between them, the main difference is their size.
Their main difference probably is that they have different cytoskeletal components, that it's a big difference between being actin only and actin plus tubulin.
Is it known
plus
whether
vimentin.
that happens because of the size?
I don't think so. I kind of think that even when we say statements like that there are two types of nanotubes and one has actin and one has actin microtubules and vimentin, even that is on shaky ground. That there is so little literature on these structures connecting cells, and the literature that there is often weak.
Yeah, and these, like, the other thing I wanted to ask about is, like, this isn't some leftover bridge from cell division. This is something that formed with two adult cells.
Yeah, great question, right? Because, like, I don't see a video here. And generally, like, I picked this paper because there was a lot of claims made about these tunneling nanotubes where I don't see a video backing that claim up. I don't see a video of one of these protruding into another cell. The canonical view here is that, yeah, these are not due to failed divisions.
Okay.
That these are things that have protruded into each other. And it looks like that, right? There's many little thin strings.
Yeah, for the TNT2s, I agree. But like the TNT1 in figure 1A right side.
I feel that I really cannot tell the difference very easily between TNT1s and TNT2s just by looking at them. I kind of reject.
They have these arrows. It says
Yeah,
TNT1.
but if you were just like, so I have some data of the chicken heart developing on the 4th day.
Right.
And in that data, you can see these thin strings attaching some of the cells on one tissue to some of the cells on another tissue, which makes me think, by the way, that it's really not much to do with cell division, that these cells divided from each other a really long time ago. I have those strings. Now I have to categorize them into, I have no imaging of the tubulin or vimentin or actin. So now I have to just magically categorize them into TNT1 or TNT2.
I think you can, right?
Right, and that's why I'm saying I can't like easily visually tell the difference between these two types of nanotubes.
Okay, but in 1A, they show TNT1, and in 1B, they show TNT2. And TNT2, I can clearly see, like, I don't have an obvious way that that's cell division.
Yeah.
A mess up of cell
but
division.
TNT1,
but T1,
you're saying,
I'm like, I guess the argument I would maybe pose is that there's more than one coming from this one cell.
Okay, so go to figure 3A1, and there's a TNT1 there. Do you see how that A1 could possibly have formed from cell division?
I don't know.
Yeah, so I think
it's obvious. Yeah,
I'm not going to like completely rule out the idea that some of these can form from cell division, but the geometry doesn't really scream to me that's what's going on.
But yeah, then if they're not forming through cell division, like let's have a movie of one forming.
Yeah. And right, like they say in this paper that they're only able to take like 10 to 20 minute movies, which I'm surprised by.
What did you think of figure 2?
Yeah.
So this was a really weird paper to read because they have such beautiful data, such beautiful movies, and just no text. I mean, the text is just like, our laser power was 2%, and this is the detector we used.
It's one of these things where you're like, well, at least this wasn't written by AI.
Yeah.
Then the figures are, there's a figure 2 in a paper filled with beautiful images and videos. Figure 2 is a flow chart that you might expect.
That you can't follow.
If you were selling like some shitty condo, this is the flow chart you would show them, not in this beautiful microscopy of mitochondria picture. I looked up, once I got to this figure 2, I was so jarred by this. I wrote as a comment, the figure is so bad, it makes me wonder if the communication in this paper is not a language barrier problem, but something about the authors. And I went and I looked up the authors because what I was wondering is the first author or is many of the authors undergrads? Which I thought would be a really good explanation for what this paper was, that this was like some undergraduate project.
Okay.
Not even close. The first author is a woman with gray hair. The last author is a guy with gray hair. These are not young scientists. So then, second theory. This is a paper written by Sherry's. Sherry is an incredible scientist. She does incredible work. She generally doesn't write papers. And I imagine if she did write a paper, this might be somewhat what it was like.
Yeah.
And so that's my current idea, is that this is a paper by career scientists who don't normally do this front-facing communication stuff.
Yeah, that seems like a good bet, like if an evangelist wrote a paper.
Yeah, exactly. A lot of the writing is tips on how to use the Stellaris Leica to get slightly better images.
Yeah.
So they would say something like, in order to reduce significantly the light exposure during multi-labeling experiments, several spectrally close fluorochromes could be illuminated with a single excitation wavelength, detected in a unique emission band, and separated according to their distant lifetime values via lifetime dye unmixing mode.
Yeah.
Okay, I followed that one, but is that true? To me, that seems like you're probably going to end up dumping more light into the system because you didn't hit each fluorophore on its optimal excitation. So you're hitting everybody off peak. So you're going to actually heat up the fluorophores more. If this statement is true, I want that shown.
Well, I think the idea is instead of having to hit it 3 times with 3 different lasers, you're hitting it once with one laser.
But let's just hit it with 3 different lasers with 1/3 the power on each laser.
So the idea is you hit them with one laser once. There, it's all pretty close to the peak.
But
and you're taking the lifetime
always
of the...
be better to hit with 3 separate lasers at 1/3 of the power on the exact right place. I don't see how that could ever be worse.
Okay, I don't see how it could ever be better. Because like a lot of your power is getting lost to diffraction through the sample, right?
Sure. That's an interesting point.
So I don't think that hitting it 3 times with 1/3 of the power, you're going to get the same signal.
You're still putting 1 watt into the sample. You still are getting the same amount of power at the cell as previously. It's not clear to me which of these is going to win. I want some data.
Okay.
Now, there is this thing that lasers can only go so low.
Yeah.
So if you are hitting at the lowest amount of laser that the laser will stay stable at, then, I mean, that's often what we're doing, right? It's like, which is why I'm always advocating for please sell us weaker lasers rather than stronger lasers.
Yeah, interesting.
Okay, another place that say, for the S1 to S0 dye transition, here they're talking about STED depletion.
Yeah.
Indeed, a dye, the beginning, they use words like indeed, in addition, as an example, additionally, like, but in the wrong place all the time. Indeed, a dye with high brightness and very compatible with STED imaging will require less excitation depletion laser power, leading consequently to higher photostability over time and longer imaging time. Again, this seems like actual advice that if you STED deplete a dye, it will then become higher photostable and you can image it better. Again, I don't know if that's true because when you STED depleted it to begin with, you dumped a bunch of energy into that dye. That's why you STED depleted it. So taking that into account, is this still better? But I felt like I was talking to an actual French person in Nancy who I was failing to communicate with. This sentence is so hard for me to process. And I have like huge block paragraphs that I don't want to read out that are like that, where I'm like, this is a lot of details.
Yeah, it's a lot of details. It's hard to follow a lot of the details. I could imagine that if you are trying to do this experiment, then like maybe this becomes a helpful paper. But the thing that really frustrated me about this paper was like they did the experiment. They had the beautiful microscope. They have it. They have the cells that are doing the nanotube tunneling, and then they do nothing with that data. And it's like, maybe this data is now publicly available, but like nobody else was there to be like, oh, like, let's see if we can see one of these form. And like that, it just feels like such a missed opportunity to try to get any biology out of this.
I've never read a paper more where I wished I had been able to talk to the authors early on in the project.
Yeah, they did so much. They did all of the like really nitty gritty groundwork for an incredible experiment and then didn't do the incredible experiment. It was so frustrating to me.
And the reviewers, I read the reviews to this paper as well, also complained about the same thing.
Okay.
And I guess maybe these people just didn't care, or they didn't understand, or
they were just like the time was up and they had to move on. I get like I understand that science isn't always perfect, but I was deeply frustrated.
A lot of the main paper, the first half of the main paper is secretly a review. Not so secretly. The first half of the paper is a review paper. We've encountered this before in papers we've done for Journal Club. We've done this one where I joked that it was ghost written by diatoms because there was so much review up front. This is on a different level where like literally this is a review paper.
Well, it has review citation formatting. So it may be something that the journal, I mean, this is the first time I've ever read something from Life Science Alliance. So maybe this is a journal format thing.
Then it switches to methods, essentially, even though it's formatted as results. And then the discussion, if you went through the discussion, is actually partially the results. That three really interesting things are shown in the discussion, which are, intriguingly, an unusual Nile red structure with an accordion-like shape was observed in TNT1, whereas staining was linear in TNT2. Wild.
Didn't they show that earlier? I think they showed that earlier.
They better have, because it's in this sentence that reading is in the discussion.
Yeah.
They also saw, and this definitely is in the earlier, the average speed of puncta in TNT2 was 6 times more than TNT1. And finally, they say, in contrast, TNT2 only contained actin and were virtually devoid of mitochondria, which I thought was interesting. So this idea that mitochondria can surf on the actin via a mirror-1 linker with the myosin, they aren't really observing that in these.
I mean, the TNT2s look too small to me for mitochondria.
And so
especially when you look at those movies, like it looks like the mitochondria just barely in their most tubular fit through the TNT1s. So
this is why I wanted to do one that was mostly data, because if you go back to the other papers, they seem quite confused on whether mitochondria, or whether these things are actin only and mitochondria are going on actin, whether the actin has nothing to do with it and the mitochondria are going on microtubules. And that's why I wanted to have something more raw like this.
Yeah.
Because the going straight for function and straight for how does this affect cancer cells and stuff like this makes the underlying cell biology and molecular biology deeply muddled. And so you end up with like right? This happened for liquid-liquid phase separation as well for the P granules, where the main hero of the story for a long time was a thing who you had no idea what the heck you were studying, right? Yeah, I wanted just the raw data rather than to get bogged down in that. They do say, and I love this, right? Because again, this is a paper that's mostly like we have this awesome microscope and we put something cool underneath it and this is what we got. Last sentence of the paper, in addition to tunneling nanotubes, broader cell biology topics could also benefit from these advanced light imaging approaches that preserve living samples. AKA, the Leica Stellaris 8 will really help people take images.
For like some of the interesting data that they got and didn't unpack at all, one thing I wanted your opinion on was the MitoTracker Red lifetime data. So did you know that MitoTracker Red would have different lifetimes in these different metabolic states?
Not only did I not know it, but I tried to do this experiment during our demo and I did not observe it.
Okay.
I was hoping that would be true, but I did not see it in our system. Now, I used a bunch of MitoTrackers, and I assume that I used MitoTracker Red, but it could be that I didn't use MitoTracker Red, right? Because I've got like 20 of them, and I tried to use as many of them as I could during the very short demo. It also is, right, a very different kind of system. I was doing a whole lung.
Yeah.
Right. I was doing mice and chicken lungs, whereas these are just plated cancer cells. Much easier. They also say, though, that because they got different lifetime values for the MitoTracker Red with their fancy FLIM microscope, that this suggests metabolic states.
Well, they say suggesting metabolic states they got different.
Yeah. I don't know if that is well known that is true. You do think when you see different fluorescent lifetimes of different fluorophores, that suggests slightly different states.
Local environments,
local environments or different states of the protein or the chemical or the dye, right? But that does not necessarily mean different metabolic states.
Yeah.
And so that's quite a leap to take unless it's been studied extensively. They don't give a citation, and I'm not aware of that being known.
Okay. Yeah, it would be, I mean,
I do hope it's true. I was looking for it at one point. That in and of
itself is so, would be so cool.
Yeah, if they could just quantify that. Yeah.
I mean, or if somebody would take the time to be like, okay, it seems like MitoTracker Red, to characterize this dye, it's like a thankless job, I'm sure, but it would be to be able to see the different metabolic states of mitochondria and be like, oh, this one is doing more ATP production and this one is producing more iron or whatever.
Yeah. This is something that I would love to do. I mean, this paper kind of sets the stage. But, and we've talked about this before, you need money to do it.
Yeah.
That there are brilliant, cheap experiments, but you really have to think quite hard. And then if you are one of the golden children who gets $1 million, $2 million, $3 million to start your lab, you are so powerful those first few years. Or you can be. It depends on how you set yourself up, right?
Yeah, I mean, this paper really made me reconsider whether it's worth it to buy something like this. I think these are beautiful experiments and so easy to do, and there's so much to unpack here. And you could definitely do this with either of our systems using this Stellaris 8 Falcon. On the other hand, investing so much of your money into a piece of equipment really locks you into your questions.
Yeah.
And that's kind of scary to me because I feel like one of the beautiful things about science is being able to pivot and try something new. But that's one of the realities of starting a lab that hasn't really hit me yet is you do have to commit to your equipment early on. This is your startup is the biggest piece of capital you get, right? So you have to buy the big pieces of equipment before you've even started asking the questions.
Yeah. And even if you're building your own equipment, like the parts that like if you want to do FLIM, you have to get FLIM equipment.
Yeah.
I think something interesting that Noah says is that it's so hard to manifest just one thing, right? You have all these scientific ideas, you have all these truths, there's all these interesting things to go after, but it's so hard to bring one fully to fruition as a beautiful paper that will stand the test of time, that you're not just locking yourself into some equipment early on. You have to really lock yourself into particular questions very early on as well, even though there are many things you could do.
Yeah.
And those are both quite painful, because we're in this because we're curious people, right?
Yeah.
And you have to a little bit kill your curiosity in order to make progress.
Yeah. And it's not something we've really had to do yet. We've been very constrained by the limitations of the equipment around us.
Yeah. But we've never gotten the option.
Had the reins ourselves and had to make these decisions up front before starting questions.
But seeing this kind of data, this is the data that I want.
Yeah.
This is the thing that I want to do. I know there's many different ways to do things. And we were talking to people about light field microscopes, and that seems super cool. And there's all sorts of super cool ways to do things. Fluorescence lifetime, specifically 2-photon fluorescence lifetime, is my pie in the sky thing I want. And just seeing this data and being like, oh, they could have done this, they could have done that. They could have taken the lifetime changes in mitochondria just on their own and see, does that actually correlate with changes in mitochondrial state?
Yeah.
And thus, can we use the fluorescence lifetime of this dye to visually image mitochondrial state? That would have been huge by itself. Like each little bit would have been huge by itself. The twist, Did you have to Google what quator meant?
No, I didn't even notice that quator was in the paper.
They have a section titled The Quator. Sample, probe, instrument, and image processing. Those are the 4 parts of the quator. Is a guideline to propose innovative tools. Quator means quartet in French. And they didn't even bother.
The thing that really bothered me in this paper was STED nanoscopy, which I assume is just like a cutesy way of saying super resolution microscopy.
It is quite cutesy, right? Because it's microscopy and now it's nanoscopy.
Yeah, I get it.
But then if they're going to make that decision...
It's confusing.
It's confusing because they've made, like, you can call these tunneling nanotubes. You can also call these microtubes. Yeah. They do state the nanotubes are between 15 and 60 microns, depending on what kind, which is, by the way, huge.
Huge, yeah.
I mean, is that right?
That can't be right.
That can't be right. What are they talking about? Why did I read that? 1 second. TNT1 were relatively long. Oh, I completely misread this. When I first read this, TNT1 were relatively long processes, 40 to 60 microns, containing actin, tubulin, and vimentin. I assumed they were using the word long wrong, because they're using processes wrong, and they meant wide, and that these were 40 to 60 microns wide, which is obviously wrong, right? Because a cell is only 10 microns. Short TNT2s, less than 15 microns, only contain actin. So
they're talking about the length between cells.
They're talking about the length. I thought the whole time they were talking about the width.
No, the width, I mean, from the scale bar is 2 microns. The width is, I would say, diffraction limited.
Yeah. Okay, so they are nanotubes. A different paper calls these microtubes, which again, I think that these somehow just didn't get that much steam, that like biologists were not so jazzed by it. And it's making me think a lot about why do biologists get super excited about certain things? Like they are not that excited about tunneling nanotubes. They are not that excited about the interstitium. They were super excited about liquid-liquid phase separation. Mid. Yeah, I think they're becoming mid. They were very excited and they're becoming mid. And I think the reason why they're becoming mid and why they weren't excited about tunneling nanotubes has to do with this critical function. That if you want a biologist to be super excited by something, you have to show, here's a new thing that you haven't seen in the cell before. And if I take it away, the cell dies. Yeah, I think the thing here is like, it's not clear when these... Like this cell line is not a typical cell line, H28, right? It's a cancer cell line. When do these normally form? Like what could be their normal function in a cell? Right, but they do seem to be ubiquitous. That we're seeing them, I'm seeing them in tissues, right? Which I think that has not been published yet. Has not, like that's not known, right? I think that will get biologists excited about it. If this is a normal developing tissue and you see these nanotubes and you can show that, then I think people get more excited. But here I can see them being like, this is
a phenomenon, but like also you can give a chlamydomonas cell hydrogen peroxide and it makes a really big pyrenoid. Like, what? Why do I care?
Well, I mean, yeah, like I would argue you care because you want to understand the rules of molecular and cellular biology. By understanding the rules of the game, you can then play the game. And if you're trying to play the game without understanding the rules, you're going to lose. But that's a very physicist perspective, I think, unfortunately. And the biologists really want to play the game.
There's so little known. There's so few rules known that, like, why are you starting with, like, offsides when you should be starting with get the ball in the net?
But, yeah, because you have to get all the rules. If you only know get the ball in the net and you try to play the game, you're still going to lose the game.
I disagree because it's an order thing. If it's a nth order rule, the likelihood that you break that rule is so low that you can understand most of what's going on in the system without ever having learned the rule, right? Like I can watch a football game, even though I don't know. I mean, I'm not even going to be able to come up with a random rule.
But if you play a chess game, and you know almost all the rules, and you don't know like 2 or 3. Like you don't know castle, you don't know en passant, you don't know how one piece moves, right? You're screwed.
Sure.
There's no way you're winning that chess game. And so I think...
Yeah, you can miss critical rules. That's a problem.
So right now, there's this thing of like, if I want my name in lights in biology, I want to win the Nobel Prize in biology and medicine or whatever the hell it's called, right? If I want my name in lights... Studying tunneling nanotubes will not do it, right? What will do it is I have to find a thing that nobody's seen before, and it has to have critical function. When you get rid of it, the cell has to die.
I don't know if that's true. Like, if you want your name in lights. Because Clifford Brangwyn has his name pretty high up there, and it has yet to be shown that these...
But it's pretty close to having critical function, right? If you get rid of, like, here's one way to think about it. There were many organelles that were known to have critical function already. And then Cliff was able to say, those organelles that already have critical function, those are liquid-liquid phase-separated organelles, right? And so you could immediately latch onto, here's a thing that I've discovered that is a refinement of something that we already knew had critical function. And that's, I think, why there was the hype about liquid-liquid phase-separated organelles to begin with, and why it's dying down now is because people were like, oh, critical function, yes. And then they were like, well, but is there anything new here? Is there any? And now your work is saying, hey, look, we could really use these rules of the game to really make some progress. But if you have something like the tunneling nanotubes, even at this point, if you find critical function, they've been around for 20 years, so people will kind of hold their nose up at them. If I want my name in lights, the discovery I have to make has to have critical function from the get-go. Or sorry, if I want to guarantee that I have my name in lights, because you never know what's going to capture the imagination of people.
Did you look up any of the TEMs or cryo-ET of these nanotubes? Yeah,
I tried to. I mean, I went through or combed through 3 or 4 papers before choosing this one. And most of those other ones were still images. Cryo-EM pictures.
And they show mitochondria inside the nanotubes?
No, but I never saw anything that I thought was interesting. Okay. I mean, let me put it this way. Let's say you're looking at William Conway's beautiful pictures of microtubule and kinesin.
Yeah.
Unless William Conway tells you what the hell you're looking at. Yeah. Do you know what you're looking at?
Yeah.
So any paper that shows me TEM or cryo-EM is, I don't have William Conway with me, so I don't know what I'm looking at. Right? Yeah. And that's true for essentially anything. The pyrenoid, and I think chlamydomonas is a bit of an exception because the structure is so massive that you can start to identify things with your own eye. But when we're talking about microtubules, I've taken pictures of microtubules with cryo-EM. I've done the experiment, and I still have difficulty identifying them with my eye.
Yeah, I would be interested. Like, I didn't get a chance to look at the data, but yeah, one thing that has been on my mind since the CMU visit and previously because of the tubules that run through the pyrenoid is like, how do you get really high curvature membranes and what stabilizes that? And so it's interesting to see that these nanotubes form and think about whether their biophysics of formation is similar to that of the biophysics of the formation of the pyrenoid tubules. And then there's also like the ER has all of these like beautiful membrane structures. And so like, is this using the same machinery to create these tubes?
That really captures my imagination about biophysics machinery here is that one type of these tunneling nanotubes has a twist to it. So it's like a Twizzlers, and the other is not.
This is this Niles red stuff.
Yeah. And you can see that pretty clearly in the images. I'm not skeptical of that claim because they pop up pretty clearly in some of the movies and some of the images. And you could start to imagine, like, we know that microtubules have a twist to them. We know that certain intermediary filaments and microtubule interacting create certain interesting things.
I mean, you definitely can see a twist in the tubules coming into the pyrenoid. There's a very clear twist.
Yeah, and so I'd be super interested to study why do the TNT1s have a twist and the TNT2s don't when they're both bilayer membranes of the cell on the outside.
And what stabilizes them to make these? I mean, maybe they're under tension.
They've got to be under tension. If they're not under tension, then I deeply don't understand.
Yeah.
But one thing I was quite frustrated with this is what I really wanted.
But then they would pop apart at some point, right?
Maybe they do. Maybe there's a snap event.
Do you think that this Stellaris 8 also has a laser ablation system?
I don't. I really don't. And but I think if you, again, all the bells and whistles in the universe, I really think that actually you have to build it. Because I've seen people try to do laser ablation with a Stellaris 2-photon system, like a dive system, and it often doesn't look super believable. I think if I'm ever going to believe laser ablation studies, I want the person doing it to have built their own laser ablation.
Why?
Because I see all these laser ablations all the time that are secretly just cooking the sample. That you're just bleaching, and then you've cooked the sample so that the proteins all dissolve.
Isn't that just a problem with laser ablation as a technique?
Well, no, because laser ablation, if you do it right, is this nanosecond pulse where you heat up the water so much that you cause the water to microboil.
Yeah.
And you get these little pockets of vacuum. The water collapses back on the vacuum, and that collapse causes a little bit of plasma, and that little bit of plasma is what just completely disconnects covalent bonds, right? Mm-hmm. And so it all happens within a nanosecond. You don't really cook the proteins, you shockwave the proteins into snapping apart their covalent bonds. And then you watch the few milliseconds
what do you get from building rather than using you're saying that the commercial setups are not doing it at a high enough power?
Yeah, not a high enough power, not a high enough speed. Yeah, both.
Okay.
They're just not built to dump all of the power in an ultra-quick chirp into one place. Okay. Even the 2-photon setup is only meant to get 2-photon fluorescence, and that it really is optimizing for a quite different problem to get clean, true ablation.
That there isn't a optimized laser ablation commercially available.
I don't know. We talked about buying one in Dan's lab once, and we rejected it. I watch a lot of laser ablation videos.
Sure. I definitely agree.
Where I'm very suspicious about what they've done.
I am not suspicious because of the hardware. I'm suspicious because it can be hard to laser ablate things.
Yeah.
And if your laser power is not high enough, you could trick yourself into thinking you ablated by just bleaching and boiling, right? So if you don't have enough penetration and you're just you can.
This is the first paper of my PhD is using High-End's laser ablation system. I took a sample that I thought was dead of Peter Foster's and tried to cut a bunch of microtubules of Peter Foster's just to prove to myself that I could do laser ablation. I did my samurai cut, and then I took a video, and instead of seeing the microtubules float away, it became very clear that I had actually just bleached the microtubules, even though I had used High-End's incredibly fancy system. And also that Peter's system was not dead, that the microtubules were still moving. And that became my and Sebastian's Nature Physics paper.
Yeah.
And there I was using very good equipment with people who really knew what they were doing. And I still didn't actually ablate.
So I feel like that, to me, I'm more skeptical. I'm not skeptical of the hardware, but I have never used a real laser ablation setup.
But it would be great to measure how much tension these are under.
Yeah.
And if they do ever pop. Another thing that I was quite disappointed at, and again, they say that they can only do this for 10 or 20 minutes. They say that the microtubules, they said
they only did it for 10 or 20 minutes. Did they ever try to go longer? Because I didn't get to go through all the supplementary figures, but they were like, it doesn't really seem to be bleaching. They were like, it goes down by 10% over. You gotta over 10 minutes.
Assume that with these people, I don't know what I assume. This is one thing that I didn't get from this, which is the whole reason why I picked this paper, is I wanted to see a mitochondria come up to a nanotube enter the nanotube, go across the nanotube into the next cell, and exit the nanotube. We
don't get to see that.
We never get to see that.
But we did. Did you watch the video of the purling?
Of the purling?
Yeah.
Oh, I did not.
I mean, they don't talk about it but it's associated with figure 4B.
Oh, very cool.
That there's a purling a purling that happens, and then there's a mitochondria that zips by, and it then gets split apart, and one mitochondria goes one way, and the other mitochondria goes the other. It's just a beautiful, that was my favorite video.
Yeah, so if you're listening to this, you might want to check out the YouTube channel for once, or the Patreon, that we put videos up on there that are the videos from the paper, and this paper was particularly chosen for some of its beautiful videos.
Yeah, you can kind of see the purling in figure like B5, B6, B7, B8. These little.
Which microtubule? Which mitochondria am I looking at?
Figure 4.
Yeah, I see B4, B5, and B6.
Yeah, you see the red arrows?
Yeah.
In B6. If you go a little up from those, there's these two little nucleoids. And when you watch the video, it's very clear that that's a purling.
Oh, yeah. Okay, yeah. Cool. Very cool. I was wondering if you thought a little bit about, this is something that you used to have in your job proposal for becoming a professor, which is that you were going to have many, many fluorophores, and you were going to separate them not only by their spectra, but also by their lifetime to do machine learning of, 20 different proteins at a time and learn what's important.
Yep.
Did you find anything beautiful about the way that they separated out fluorophores here? Or did you find anything informative about how you would do it?
Well, they did. They cited a paper. Interestingly, a recent publication has reported simultaneous imaging of 9 fluorescent proteins in a single acquisition using FLIM combined with interleaved excitation of 3 laser lines. And they cite a paper from 2023 there. So people are making progress on that. What was the question? Is there anything beautiful?
I was wondering if you had gotten excited about the techniques they were using in this paper. I'm surprised that one of your takeaways from this paper was, if you buy this microscope, you've dumped all of your money into doing this specific thing. Because for so long you were so excited about doing a project that's similar to this.
Well, this is why, I'm not saying I wouldn't dump the money in, right? I really these are beautiful experiments, and it's such a powerful technique. This single microscope could be the only microscope I ever use. It can do all of the normal microscope things. Like you can image clammy live. I can look at my condensates. I don't think I can do FCS.
Right, you can't do FCS.
Yeah. And so, this led me to the question of what do I really want to study, right?
And again, actually, we talk about this single microscope. An older, shittier version of this microscope, me and you, the one paper we ever wrote together, was literally on an older version of this microscope. Yeah, you can
hack this microscope to be able to do FRAP. So this can do a lot of things, and it would open up this project, which I still think is an interesting project with the right student.
But there's always things you can't do. You can't do FCS. You can't do ultra fast imaging. I mean, I would argue that this thing is particularly bad at fast imaging. I felt that was a real weakness of it.
Fast imaging. Yeah. So if I want to look the fluctuations of droplets, that's going to be not possible to do. That I could do on a wide field, very cheap microscope. But I did feel like there's a decision point here of do I want to be thinking about the teeny tiny scale and looking at FCS type measurements of individual molecules, or do I want to be doing FLIM? Does that open up more doors? Because both of those are very high priced items, and I'm not going to get a startup that allows me to buy both.
The decision that I always feel when looking at this particular microscope, because this is the dream microscope, right? This is the Lamborghini you dream about, right? The decision that I always think about is, do I want to buy this?
Or build it.
And the things that I go back and forth is, do I think I can build a better version of this?
Yeah.
Yes, I do. Although there's a big asterisk, which is
white light laser.
White light laser. And the way that the Leica detectors work is hard to compete with these days.
And also the analysis stuff. I mean, I guess with ChatGPT, this is all going to get a lot easier, but it's pre-built in and it's already validated and stuff that I think can't be undersold.
And so I think I'm good enough in making analysis packages. This is something that I really excel at. But whatever I make is going to be totally unusable to anybody not in the lab, not being trained by me.
Yeah.
And if you want to be a part of a community and do all sorts of exciting things, this microscope is a really good way to do that. Where building the equivalent microscope is a thing that only you and the people that you train will ever understand how it works, because it's such a complicated beast.
Yeah. Interesting.
Long advertisement. But the paper is a long advertisement for this microscope. So I'm glad I've used it in my life before, so I can have...
thanks for doing that demo. I have also gotten to use it. And we got to separate out, like, this is why I think it's such a cool system for some of my questions, which is that Chlamydomonas has chlorophyll, and that separates out from everything super easily. And so you can have all these other fluorophores. One
of the things that they do early on in the paper that I actually didn't understand, I mean, there's a lot of things they do, is the way that they take their images for figure 1 is they have Alexiflora 594 and Atto 647 vimentin and tubulin. How are they getting these pre-conjugated vimentin and pre-conjugated tubulins into the cell?
What is vimentin?
Vimentin is an intermediary filament.
Okay. You don't think they're doing some labeling?
Well, they've pre-conjugated. Let's just talk about the tubulin, where you know it. They pre-conjugated.
Oh, this is fixed. 1A is fixed.
I don't think so.
Yeah.
I don't think so. Why do you say it is? At first, I thought it was. I had a question mark. Isn't this mean that they're doing fixed?
They said they were doing fixed in figure 1.
Where?
Let me find it again.
That would answer my question, right? And that's what I thought originally. But then I thought about it again. Like, that would make, for instance, using the 48 conjugated phalloidin make a lot more sense, that you've used the phalloidin and
We started our study by conducting triple labeling through immunocytochemical experiments.
Yep, I read that.
Use it in fixed H28 cells.
Where does it say that?
In that same sentence.
Yep, I see it. All right, good job. That makes a lot more sense, because otherwise I didn't understand how they did this. Because there are like tricks to sometimes get pre-conjugated tubulin into your system or something like that. But I didn't see how they could do it here.
They started in fixed, and then the rest is in live.
I'm surprised a little bit that you can capture this structure. I mean, I guess it's important for all the TEM and cryo-EM, but it's a small structure to capture in fixation. You imagine that there's a thin little bridge between two cells.
Yeah.
And then you add paraformaldehyde or something to the sample. All the cells shrink by 50%, and that doesn't rip this bridge.
Yeah, I mean, I guess this is another reason why I could imagine people haven't been on board with them, is like, are they just artifacts of the fixation, of the imaging, of the TEM, of the cells being stressed out from that whole process?
Yeah, but it's just like, why were people then on board with liquid-liquid phase separation? When, again, if you didn't know better, you could be like, oh, this could just be an artifact. This could just be the cell stressed out. This just could be some weird form of blebbing or something like that.
Yeah, I don't know.
Yeah, some things just stick and some things don't. But this seems like a thing that I would be really excited if the community researched a bit more.
Sure.
Because this is one of these things where, like, I don't really care about what is the critical function, what is the goal scoring mechanism. I really care about the rules of the game. I want to know how cell biology works. I want to know how molecular biology works. And this seems like a giant question mark. And there's plenty of giant question marks that people should go after. I'm pro going after giant question marks in general. This one seems particularly, it's microtubules and mitochondria.
Yeah, it's got all the players
and twisting. Yeah. It's got all the players that I love, and I'd like to get into it a bit.
Yeah, sure. Go for it. I support that. If you could add one thing, what would you add?
I would add a longer video.
Longer video.
I picked this no. Okay. Yeah, I would. If we're going to do no physics and no biology in the paper and just have a paper that's pretty videos and descriptions of how you got it, give me a really long video. That's what I want.
Yeah.
What would you add if you could add one thing?
I want to label all the mitochondria in one cell, have it make nanotubes, and see if any of those mitochondria end up in neighboring cells.
Yeah, I mean, this is essentially what I have tried to do before, and quickly decided I didn't have the tools, including the microscope, to do it. But yeah, I'd love to do this.
I mean, I just think, like, they had it all there. They could have done it, right? They were using dyes. So they could have, like, seeded, they could have labeled the cells and then seeded very sparsely the labeled cells and then put a bunch of unlabeled cells around them. They were there.
Yeah, what a weird paper. Again, I loved it. I loved reading it. I loved looking at it. I loved thinking about it. I think that there should be a genre of just data dump papers for people who are sick of doing experiments and are just like, give me the data. I just want to look at the data.
Yeah, very cool paper. Very like cool to see this technique. We saw the microscope, and the reps will tell you, like, talk themselves blue about all the things you can do, but it's cool to see all the things you can actually do.
Yeah.
And yeah, the last question I have is, like a Stellaris 8 with all the bells and whistles, worth it?
I think, again, it depends on what you're doing. I think if you're doing thin cells that don't move quickly, then the answer is absolutely yes. It's the best microscope that exists right now. And in fact, the guy who didn't hire me, what's his name?
Morel?
No, the guy, yeah, that's true. the guy who is right next to your current lab.
Oh, Kai.
Kai has bought the sister of this microscope, which is a Leica Dive, a 2-photon Leica, but specifically without this module, which is the other side of the best microscope that you can get. And so I do, thin samples. I think that slow samples, this is the best you can do. If you want to study organs or whole creatures or something that moves fast, I think that there's better that you could do. If you want to study organs or whole creatures, you can go the way that Kai has gone and get the sister of this microscope. I would argue once you want to study whole organs, there are competing microscopes that you could also buy. But I'm of the camp, build it yourself. That when you want to go big, building is the way to go.
Yeah. I mean, it does give you the flexibility of changing your geometry much more easily.
Yeah. And then if you want to go fast, I don't think anything in the Leica ecosystem is good at fast imaging. In fact, I've talked to them many times that they have to get some sort of spinning disc in their company specifically for this reason. And they've kind of done that, but not really. It's like a spinning disc, but it's not very impressive. Yeah, if you're trying to go fast, there's a large ecosystem of other things that you would do. And then I would say about half the science being done in biology that's microscopy right now is going fast. So there is a giant swath of people where this is not the right microscope to buy.
Yeah, I see you.
But I'm not one of those people. I'm not really trying to go fast.
Yeah.
But I am trying to go big. So this is like on the
edge.
On the edge for me.
Do you want to answer the 4 Asya?
Yeah, I've got a thing to say.
Okay.
Yeah.
To the picker.
To the picker. That's me. I picked this paper.
For Asya, broadly in the world, why did we, your parents, care?
This is an inflection point of our career in many ways. We are going to have careers. Yeah, sorry. Yeah, we're two separate people. We're going to have a lot of difficulty continuing our career past this point. We always knew that was going to occur. We always knew that by getting married and having a family, we dramatically reduced the chances that we got to become professors. But this is the first year that's really become very clear that no one's going to save us. No one's going to go to bat for us. People were very happy to hire us and have us do work for them. But the idea of letting us continue our family and have 2 labs and have this kind of idyllic setup, nobody has really like incentive to do that. We might survive it, though it likely will not come from a university. But one thing that this has made me reflect on is what I did in the 15 years that I was given to be a scientist working for other people. And there's a lot of things that I regret. I regret that I wasn't louder, that I didn't speak up more, that I didn't tell people in power that they were wrong, that people pinned me very early as a loud person and used that to tell me to be quiet. And I became quiet because of that. And I wish I had spent 15 years, even if it meant I only got 10 years instead of 15, I wish I had spent those 15 years screaming at the top of my lungs what was true, what was not true, what was beautiful, what was the point, what was unethical. Because I think a lot of really deeply unethical things have been creeping in to science. One of the things that I don't regret is what I studied. I am so deeply proud of every bit of my work. I'm deeply proud of the colloidal system I found as an undergraduate. I'm deeply proud of these stress-free straining systems that we found in microtubule kinesin systems. I'm so happy that's being followed up on. I'm deeply proud of these co-block polymer-like active systems that make these active foams. It breaks my heart that 5 years later, still nobody has published a similar thing to what I published in 2021. And I'm desperately hoping that somebody follows up on it. I think people will. I've heard rumors that people will. And I am deeply proud of this paper that is coming out now, which is going to be that energy metabolism in developing organs can be spatially patterned. And we should look for energy metabolism patterns throughout the world. And maybe that will help us understand diseases. It will help us do the scoring a goal in a net version of the rules. But nothing I have done has been deemed important by biologists or by physicists. Beautiful, yes. Notable, yes. But name and lights, not even close. And so I'm thinking about, if I get one more chance, if I get 5 more years, what would be a project that would put my name in lights? Not necessarily that I'll do it. Name in lights is a pretty bad goal, right? You don't want to just do a project for the sake of getting a Nobel Prize. You don't want to dedicate your life to getting a Nobel Prize. You want to say, screw scientists and their terrible ethics. I'm going to learn the rules of the game. But I would like to be able to say in my mind, I could do A and get my name in lights, or I could do B, which is the most important thing. And that involves understanding why scientists got super excited about some things and not about other things. It involves understanding the psyche of the giant group of biologists, which collectively is making quite a lot of progress, even though individually all the beliefs that everyone is following are quite myopic. And so this has been a little dive into something amazing that the biology community has kind of scoffed at. And it's getting some traction, but it's not getting much traction. And if I talk to a biologist about it, no biologist knows what a tunneling nanotube is, just like no biologist knows what the interstitium is. And I think that by reading this and by thinking about it and by thinking about why it didn't get traction and other things did, I am slowly understanding what kinds of projects I could do that would put my name in lights and what kind of projects I can do that won't. And so that's why I wanted to read the paper. And then also I talked to my friends about it, and I didn't really understand it. So I thought I should go into it some more. But that's why I picked this specific paper.
Yeah, it's a beautiful paper. Thanks for picking it. I loved watching the movies. And yeah, it's one of these fishing expeditions. It's the thing that kills me. Yeah.
There was a fishing expedition where they were throwing the fish back in, though.
Yeah, no, that's what I mean. And it's like this would be a great, I want to say that it's like a resource for the community, but nobody has this setup.
Yeah.
Right? Like Princeton doesn't have the setup.
Yeah.
So
it's incredibly expensive. And even if you did have the setup, this is not enough details to help.
Oh, yeah? I don't know. I didn't like
I would want quantitative proof about the things that they said, such as it really is better to hit with one laser at 3 times the power than 3 lasers. Like, I want quantitative proof. If this is a methods paper, you got to prove to me that the methods you're using are better.
Oh, I don't think that this is a methods paper. I think this is a we tried it paper.
Yeah, it is. It's a we tried it paper.
But yeah, it was.
But a we tried it paper is not a resource, right?
Yeah, it's not a resource. It's not a resource, I think, partially because, it's not thorough enough on that end. And then also that this microscope is just so out of reach that, like, if anybody was going to do these experiments, it was these people.
Yeah.
So.
Yeah.
Missed opportunities.
Or, maybe they'll... how long ago was this paper? I think this paper was actually a few years ago.
I think it was
2024. I don't think that it took about a year in review, which I was surprised by, that it went through a pretty long review process. Not that, I think it's like 9 months, but something like that.
Yeah, I mean, maybe they're following up on it. We don't know. I haven't looked at this last author's website or anything.
I don't think so.
But
Thanks
a lot of inspiration. Yeah.
Send this to your friends and family.
Go check out my paper.
Oh, yeah, Linnea's paper is out
in the world.
Yeah.
It's open access, Nature Cell Biology.
Very exciting. Should we do that for our next Lemma Journal Club?
No. God.
And we'll see you next time.
Bye.